Okay, so my job is to introduce Ashwin Viswanathan. He's a scientist at Nature Conservation Foundation, Bengaluru. So Ashwin did his PhD on plant-fungal interactions. It's kind of studied, I really wanted to say that because we don't do that work anymore. So he studied how presence of fungal pathogens influence mortality or survival of tree saplings. So he studied that in the same place I did field work. Remember Cardamone, the forest plantation mosaic I was mentioning? So he did field work there. That's how we met. And currently he's working with Bird Count India. So it's hosted within Nature Conservation Foundation. They kind of use citizen collected data to analyze and infer patterns of bird abundance and distribution in the country. They recently came up with a report. Hopefully Ashwin will mention about it and you can look at it. Yeah, but Ashwin used this citizen collected data and use statistical models to get the trends and distribution of birds. So also Ashwin worked with us in understand certain patterns in pathogen data we collect. So welcome Ashwin, and over to you. Yeah. And thanks for coming today.
Hi everyone, can you hear me? I hope you enjoyed the first part of the day. I'm going to talk about simple use of statistics for disease data, but it's generally applicable for various data sets. It will be at, I know that people are from a very diverse audience. Some of you might find a few of the concepts obvious and simple. Some of you may not. So this will be at a relatively basic conceptual level, where initially I'll show you simple ways to understand data, and then some generalized linear models and regressions and how you can use that to understand this data better. So first, the scope of this lecture. First, we'll talk about the typical data types that are collected, at least in my experience, typically in these disease ecology studies. Then we'll go through some simple stats and regression and talk about how to deal with the data. We'll also get into the design, into study design a little bit, and what that means for the kind of analysis that you'll be doing. And finally, I'll show some visualization. So this will be there through the presentation. Throughout my lecture, I'll be asking questions and we'll keep it as interactive as possible. Please engage, right? And anybody who knows an answer, please give the answer. So I'll start with these different data types. How many of you have already collected some kind of disease data? Just 3, 4 people? 4, 5, 6, 7, 8, 9. Okay, quite a few. Have you collected presence absence data? Is that a sort of data that you see often when you're collecting disease data? Under what circumstances do you collect presence absence data? By presence absence, I mean a vector or a host either has a pathogen or doesn't have a pathogen. Under what circumstances do you collect that data? Okay. That's the method, but what is the organism? For what organism are you collecting this data? Okay. 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 So you see whether the fungus is present or not present. Yeah. So this is a very common data type. And this data type is actually applicable to host vectors and pathogens themselves, right? You can look at whether pathogen is present or absent in a host or a vector. You can look at whether a vector is present or absent in a host. You can look at whether a host is present or absent in a forest landscape or a community or a particular site. So this presence absence data is a very valuable type of data and for various parts of this entire ecosystem. Next is prevalence. So have you directly collected prevalence data, any of you? Yeah. How is it different from the presence absence data?
It's a gradient like 0 to 1. So like in percentage or 0 to 1. But presence and absence is like two data, 0 or 1. And by
So how do you get prevalence from presence absence?
continuous and that is
What I meant is how do you actually collect that data? How do you get the prevalence data? Let's say that you're looking at whether the fungus is present or absent in each one of your organisms, right? Where is the prevalence data coming from?
prevalence when we go to the samples, like 100 samples, we do how many are there.
Right. So you aggregate across presence absence and you get prevalence data, right? Prevalence data, you can also get more, it's sometimes more relevant when you're doing experiments. It's something I'll come to later. When you start off with a set of, a starting set of organisms or maybe 10, and then you see how many actually have that pathogen. So this is again valid across the entire ecosystem, I'm calling it, right? Then finally you have counts. Under what circumstances would you have count data in disease ecology? Anybody? Can you count a pathogen? No, right? It's tough. Sorry?
Infected, died, alive.
But that again often becomes a proportion. When do you have a count standing by itself? Sorry? Right. Whenever you're looking at host populations or anything other than the pathogen itself, when numbers become very important because it's important to know abundances of the hosts and vectors to understand what is going to happen with the pathogens they harbor. That also you can count, but that also becomes a function of the denominator. How many individuals were there? How many individuals are affected divided by how many individuals there were, which is in a way proportion, which is like prevalence, something between 0 and 1. But then you have these counts themselves as the thing that you're interested in. Counts typically are not applicable to the pathogen itself. So this is just to give an overview of the different data types. And now we'll start off with a broad hypothesis, and I'm sure Ansil has already alluded to a hypothesis in this direction, and it's something that all of us are interested in. A hypothesis is something that is untestable. So is everybody clear about the difference between a hypothesis and a prediction? Yeah? Anybody not clear about that? I'll anyway just explain what it is. A hypothesis is a very broad, untestable theory. It's like pathogen prevalence is higher when natural landscapes are modified. Now, that by itself is something that you can't immediately go and you can't modify a landscape, and it can be modified in so many ways. There are so many different ways in which landscapes change. So it's a very broad theoretical idea, right? So the idea is that pathogen prevalence increases when landscapes are modified and when new host and vector communities come in contact with each other, new pathogens come in contact with each other, right? So prediction is something that is actually testable. And that is where the next step of statistics comes in to actually test these predictions. So prediction is usually something quite specific. When pathogen prevalence in certain hosts or vectors will be greater in fragmented versus unfragmented landscapes. So that's a very specific prediction, which means you can actually design a study to test that prediction. You can go to fragmented and unfragmented landscapes. You can collect some data, which we'll come to, and then you can actually look at the difference between the two. And then the rest of this lecture, we look at how you can look at the differences between these two. This is a very typically how we do science, right? And when you have these predictions, you have a study design that accompanies that. So how will you test this and how will you go about collecting this data specifically for this? Does somebody want to say anything here? Yeah. Right. So you're actually, what you're doing is you're testing the prediction. Yeah. 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 Yes, yes. What you're actually, your hypothesis is actually something that is untestable. It's something much larger, much broader. That the way that we alter the world is going to impact pathogen spread. That is a very, very broad idea and concept. And a prediction is something more specific that is testable. And in this particular case, how will you test this and how will you collect data for this? Can somebody? So how will you collect the data in the first place? How will you design a study to collect that data right from the start? Ah, right. So you have a broader landscape. Within the landscape, you might have many sites. Can everybody make out the difference between these two colors? Yeah. Yeah. So anyway, so this is unfragmented. This is fragmented. So you have some sites that are fragmented, some sites that are unfragmented. Within each site, you have multiple different plots where you sample. Let's say you have small subsites within those sites. In each of those sites, you're actually looking at, let's say, frogs. You go and catch 100 frogs, and you see how many of them. Each one you see whether it has the fungus or doesn't have the fungus. I think the frog and fungus is a good way to go ahead here. Although I've put a mouse over here. Uh, right? So this study design is very clear, right? So then the next step becomes very easy. You actually want to find a way to look at the difference between the greens and the blues statistically. Okay. So now there is a general tendency in while using statistics to go to the most complex statistical method right at the start, right? You want to do a GLM or you want to do some complex regression or some fancy model, but actually the place to start off is a very, very simple comparison of means and standard errors. That is the first step, right? To see whether there is any difference between the data. Now this is limited. This kind of simple statistics is limited because you can't actually look at the impact of multiple covariates at a time, right? You can't, you can look at the difference between fragmented and unfragmented, but what about everything else that is different between these two sites? What about everything that is different between the organisms themselves? What about the size of the frog, the age of the frog, the, I don't know, species of frog maybe? So all of these things, right? There are many different factors that you cannot look at everything with the simplest statistics, but it is the place to start. So first, everybody has used box plots. Has everybody used a box plot? No? So box plot is, I mean, I'll explain it just after this, but first, can somebody tell me the difference between a box plot, which you use in R or any other software, you just do box plot of whatever your data is, and you get these two things that are often presented even in formal work. Right. Exactly, yeah. So that those are the right words. It's the distribution of the sample, right? Box plot is telling you about the spread of the sample. Now, is this what you want to use to compare? Do you want to look at the spread of the sample, or do you want to compare something else? What do you actually want to compare?
And then we can see that there is a biasness in sampling or that also we can see.
To an extent, yes, yeah, that you can see. But to compare the difference between fragmented and unfragmented, right? Let's say you have that on the x-axis. You have two box plots. What does that tell you? Let's say that one box plot for prevalence in unfragmented is lower than fragmented. That you have found. But is that what it is? Can a box plot actually tell you that? And what is the difference between a box plot and a comparison of means and standard errors. You've also seen that, right? You'll have a point and you'll have whiskers. You'll have a bar and you'll have whiskers. That is not the same as a box plot. So what is the difference between the two? Yes, yes. Okay. So it is actually the spread of your sample. I'll just show it in the next slide. But it is just the spread of your sample data. So typically, do not use a box plot to draw statistical inferences. It's just telling you about the spread of your data. What you're actually interested in and what all of the models and various, the more complex statistical models do is they actually compare means. They compare means and the confidence that you have in the estimation of those means. And what are those means? What are those estimates of means? It's actually, you're trying to get at something to do with the larger population. Right? You're trying to infer about the population rather than just that sample. So how would you do that? Typically what you do to calculate these means is you do some kind of bootstrapping. You take your data, which is used for the box plots, the sample data, then you resample from within that sample data by, like you have, let's say 1, 1, 2, 2, 3, 3, that's your sample. Now within that, each time you pick 6 numbers out of that, and then you calculate a mean. So you calculate 1,000 or 10,000 different means. And then you look at how precise your mean is. So based on how much variation there is in a sample, you'll get an estimate of how confident you can be about your population mean, assuming your sample is somewhat representative of your population. So actually what you're going to get is not the spread of the sample data, you're going to get the confidence in your population mean. So that's when you have the point and you have the confidence intervals around that. Whereas this is telling you the spread of your data. It's giving you the median and it's giving you the quantiles after that. So it's something like this. So this, let's say, is your sample distribution. Right? You have a box plot, you have the median here, you have some quantile around that. And sometimes you have these points lying around here in a box plot. Actually, what you want to do is you want to calculate a large number of means. So this is a histogram of whatever we have on the y-axis here is there on the x-axis over here, right? And each time we calculate a mean and we look at where the central 95% of those means are. And this will give you the confidence intervals, the lower and upper confidence intervals associated with this mean, which is likely to be very similar to this value. But what you have is, instead of having it near 1 and -1, you actually have it much smaller. You have it somewhere here. Is that clear? So the difference between these two. So whenever you want to do these simple comparisons, don't use box plots. Use a slightly more formal technique, which is calculate these bootstrap means and look at uncertainty around that. So that tells you how much confidence you have in that mean. And if there is no overlap, let's say, I'll just show in the next thing, you can be fairly confident that these two come from different populations. Right? And that is the inference you're making. So over here, going back to the blue and green, blue was unfragmented, green was fragmented. This is all made up data, so don't take it seriously. So you have the blue and the green. This is what you get with the box plot. Box plot, there is considerable overlap. There is not much you can infer, and there's nothing actually you should be inferring from it. But actually, if you look at the means and the 95% uncertainty associated with the means, here, you can see that data 2, which is the fragmented population, has slightly greater prevalence. I mean, ignore these specific numbers, but this can think of it as 0 to 1. So is that clear? So here you can look at this and you can infer that this comes from a different population that typically has greater prevalence of pathogens. Yeah? So that is the simplest way in which you can analyze this kind of data. What do you do when, so here, if you see these axes and if you actually want to take means, etc., you might actually be dealing with counts. Often you can do this for the hosts or the vectors or these abundances. But when you're dealing with a proportion, which is prevalence, let's say 5 out of 10 or 7 out of 10, and you have many different, let's say in each of these plots that we had before this, we get some prevalence value. right? Here we have 3 out of 7, here we have 5 out of 9, here we have 4 out of 6 or whatever, right? We have many different proportions. Now we want to find out, we can do two things. One is you can take the means of all of these proportions and use exactly the same method to get the means and uncertainty. You don't just have to do it with counts, you can do it with many samples of proportions. What are you doing? You're taking all of these proportions, that is one sample. Then you're taking a mean across that, and with resampling, you're getting the uncertainty around those means. That's clear to everybody, right? But you can also have the possibility of across the entire site or across the entire treatment here, which is, let's say, across all unfragmented areas, you have, let's say you have 1,000 different hosts, and in those 1,000 hosts, you have about 50 with the pathogen, right? So you have 50 by 1,000. Similarly, let's say over here, in these, you have only 50 different hosts. You didn't find so many hosts. And out of that, you have about 30. So these are directly you're getting a prevalence value, right? You're getting one is maybe some 0.05, the other one is 0.5 or 0.6. But can you then say directly that over here, the prevalence is lower than here? Again, you can look at means and the uncertainty. What you've calculated here, this 0.05 and the 0.8, by dividing these two numbers, you're actually getting a mean. But now for this kind of binomial data, where you have a denominator, you have a numerator, you can actually calculate a binomial confidence interval around that. So I'll just show that. So binomial confidence interval for prevalence. Now this is determined by this P is 0.4. In your in one particular treatment, you got a prevalence of 0.4. In another treatment, you got a prevalence of 0.7, right? In one of them, you actually had only 5 samples. In the other one, you had 20 samples. Now, if you just look at the mean values, you'd be tempted to say that here the prevalence is greater than here, right? But that is something that you should not do. You should look at the denominator and what that means for the confidence you can have in this particular mean. So there are straightforward ways to calculate binomial confidence, and if any of you use R or any other softwares, there are functions for it. You can use the function called binconf, and there are various functions that you can use. And in many of these things, ChatGPT is your friend. If you want to find out how to do it or whatever, I think Perplexity or whatever people use nowadays, there are better versions. Yeah, so somewhere I did one a small workshop on how to use ChatGPT for coding, and then at the end of it, they told me that I shouldn't be using ChatGPT, I should be using Perplexity, and then people have. I think there are so many new AIs that are coming up that apparently are better than each other. I'm sure many people here are aware of this more than me. So yeah, so just use your favorite AI, and you can get the code to do something like this. Yeah. Yes, yes, in the upcoming slides. So here, this what Farah was talking about is this n equals 5 is the sample size here, which is really critical because here you see that with the confidence intervals around this, you actually cannot make any inference about this. Both of them might actually be from the same population. That's right, no? The confidence intervals are overlapping. Let's say that if you have some a particular I mean, you can't really say that the two are different over here. But what happens if you increase your sample size? So now I've increased it to 20 and 30. Immediately, the confidence around that mean improves. That's because it completely depends upon the amount of samples you have. So if you're doing a study, don't do it with 4 or 5 frogs, try and do it with 100 or 200 frogs in each treatment. So you have this, and so you see now they're starting to separate out a little bit. And if you have even more sample size, they eventually start to separate out. Then you can actually say that these two are from different populations. You can have much more confidence in saying something about differences in prevalence in both places. So here what we have gone through is very simple statistics. In many cases, this is all you're going to need to tell a story, especially if you don't have many covariates that you've collected. If you have something very simple you've just gone looked at frogs in one place, you've gone looked at frogs in another place. There isn't any more complexity in the design than that. All you have is a prevalence in both areas, and if you want to be able to say something, this is the way to say it. If you want to be able to say something with a slightly more complex study design where you have plots, each of them you have sampled, then you might want to take means across all of those, and then you can show the previous visualization and calculate and show the previous visualization. Yeah? Okay. But now we'll come to the to how to deal with little more complex data, and yeah. Right. So box plots, again, let's say I'll give the frog example only. You have 10 different sets of frogs that were sampled. In that you get a whole set of proportion values. In one of them you had 0.1 was the proportion of frogs that had the fungus, another one it was 0.2, another one it was 0.2, one of them it was 0.3. So you have many such proportions. Now there are two ways in which you can handle this data and show it over here. One is you can just look at how much variation is there in all of these proportions you have got. So that is what a box plot is. It actually tells you the variation in the sample. What you're doing over here with means and standard errors or confidence intervals is you're then taking a mean across all of those proportions, then you're resampling from that, which means you have these 10 or 20 different proportions. Within that, you're selecting another set of 20 proportions from that sample. You're calculating a mean again, and you're doing this whole thing 1,000 or 10,000 times. Each time you're getting a slightly different mean. Now, if there was a big difference in the sample, if it went from 0.1 to 0.9, if you had 5 values that were 0.1, 5 values that were 0.9, actually you'll find a lot of uncertainty in your mean. Because some cases you'll get everything as 0.9, some cases you'll get everything as 0.1, and your mean, these 1,000 different times you resample, you're going to get wide variation in the means, which means you're not going to be able to actually say anything, you're not going to be able to infer anything. All you can infer is that you don't have the you don't have the evidence to say anything. Rather than, you have the right data, but you don't have the evidence to make any further inference, or you don't have the evidence to say that there is greater prevalence in one site versus the other. But for the binomial confidence intervals, I came to that where you actually don't have multiple samples. You just have one sample where you have 0.1 is the prevalence, and you have another sample where 0.6 is the prevalence. How do you compare just these two? And that is what I showed after that. Because sometimes this is what you have. You don't have multiple samples. I don't know whether anybody has actually faced these situations, but you can have either of them. If you have a very well designed, carefully designed study from the start, you may not have to do this. But if you don't, if you just have two sites, you have randomly collected in both, you just have a prevalence in each, then this might be the way to explore it with a binomial confidence interval. But now we've spoken about something very simple that has ignored every other aspect of biology that can explain disease, right? We have only looked at the simplest comparison, which is fragmented, unfragmented. We have forgotten about any morphological features of the organism. We have left out any habitat covariates of the site. What about all of that, right? And that is where we want it's useful to move on to regressions, where you can explore multiple different covariates at once, and you're not restricted to what a very simple unidimensional visualization can tell you. Now we can get into something more complex, and now we'll discuss that. So some of you have used GLMs. I'm going to explain all of this after that, but a GLM is a generalized linear model. It is the same, conceptually, it's the same as a linear regression. What do you have in a linear regression? You have y is equal to some ax plus b, right? You just have a linear equation. You have your response, and you have a covariate which has some number associated with it, and you have an intercept. And what do you do in such a case? You try and find the best fit line that fits your data, right? That is what your linear models are doing. So you're getting this, if you have y is equal to ax plus b, what is it estimating? It's estimating this a and B values that will best fit your data. But this is for a linear regression. What do you need for a linear regression? You cannot do that with binomial data. You cannot do that with proportion data, right? You cannot even do it with count data because none of these data are normally distributed or follow a Gaussian distribution, which is like this. You see this. This is a typical kind of data that is relevant for a linear model, a very simple non-generalized linear model, right? I mean, here we have some bootstrap data which is for mean, so not for that. But if your data, whatever it is, like let's say the length of a leaf or the size of a frog or the weight of a rat, many of these things, if you look across the entire population, it's likely to follow a Gaussian distribution. You'll have a mean and you'll have a nice smooth curve around that mean. But what do you have for presence and absence? Or for proportions, they're bounded. For proportions, it cannot go below 0, it cannot go above 1. So what if your mean proportion is 0.05? It's not going to be uniform around that mean. Is that clear to everybody? I mean, some of this will become clearer further down, but just at this stage, conceptually, you have your 0 and you have 1. If you have a proportion that is closer to any of the ends, you cannot have the same amount below as above. So if you look at these binomial confidence intervals here, one thing you'll notice is below the point, the uncertainty or the confidence interval is actually greater than above, right? And that is an inherent part of proportion data. This kind of data, you cannot use a simple linear model. This is where you need a generalized linear model. And if you over here, like imagine the point is somewhere here, you might actually have a very tiny upper confidence and you can have a much larger lower confidence. So then what is generalized? What is a generalized linear model? What is happening? I'm sure many people here know, but does anybody want to answer this? What is the difference between a generalized linear model and a linear model? What about a generalized model makes it appropriate for this kind of data? Or even for count data, something that is not symmetric about the mean. Anybody? Yeah, just go for it. Like, what does logic dictate? Anyone? Yeah, yes.
Which is a very which follows a normal distribution. So GLM will have the data which falls under a non-normally distributed data.
Okay.
So this is a major difference between two, and
so that's about when we use these linear model versus GLM. But what actually happens in a GLM that makes it suitable to look at non-normal data?
okay, so this
That is the question.
generalized is the one which will be after using GLM, the data will be normalized, and then we can see the difference in that.
Yes, I mean that's approximately what I was looking for. The data is transformed.
Yes.
So you keep hearing these words like transformation, and you have so what happens in a GLM is that whatever your data type is, there is a certain function that is used against that to make it normally distributed. So if you have count data, you typically do you have a logarithm of that value to then make it more normally distributed. So what you're effectively doing is you're doing one particular operation on your response, like whether it's a count or a proportion, you're doing one particular operation on that, so that whatever you get out of that can then be treated like a linear model. So that is effectively the difference. But there are many other like associated dependent impacts statistically, which makes it which requires so the different ways to interpret it. So that we'll get into in a bit, but the main difference is just this, There is a transformation done to your non-normal data that makes it normal. So that is what a GLM is.
If our data is like continuous, but it's not normal, then also we can do that.
Yes, yes. Yeah, yeah. You can. And the nice thing is there are inbuilt ways to be able to do this easily. You don't actually have to do this transformation yourself. You don't have to figure it out. There are certain, now with GLMs and with all of the advances that we have in statistics nowadays, you can, with just one statement in your GLM function, you can tell it what you want. Huh.
When you ask questions, nice if you ask using the mic. So probably others also will have the same questions. So you can hear and get them clarified. Also, the sessions are recorded. So if you have questions, raise your hands, and some of us will get a mic.
Huh.
Next time.
Yeah, sure. Okay, so there are different kinds of transformations that are used for different kinds of data. Like for presence absence data or a proportion, you use a binomial GLM. It's binomial data, and the reason I call it a binomial GLM because now things are easy. You just have to write a simple thing in your code. You say family equals binomial, which will show that is all you need. You have binomial data, you don't have to get deep into the mathematics, you don't have to really do much of the you don't have to do the transformation yourself. This is going to happen within the model statement. Next, if you have count data, you typically want to use a Poisson GLM, which means there's going to be a different kind of transformation and there's a different way you want to mention this within your GLM. I'll show how you put out a GLM statement, how you write a GLM statement later. And then you must have heard of a GLMM or a mixed model. So now that's one step beyond that. So can somebody tell me when you would use a GLMM or when you have mixed models or when you have mixed effects? So you have fixed effects and random effects. I'll get to when do you have random effects? That's when it becomes a GLMM. So now does anybody want to guess? What is this extra M? Yeah, it's about trying to interpret what is written here as well.
So
Yeah.
there are certain predictor variables which we might not be interested in, but they can influence the results.
Yes.
So those are the random variables which we should account for even if they're not part of our predictions.
Can you give an example?
It's usually like space and time because these things inherently can affect your results when
Yeah.
you collected the data or where did you collect the data from. So they are usually the random.
Yeah, that's right. It's something where you actually don't have a measurement to be able to tell the difference. If you have different sites or you have different seats, you're all sitting in different seats. And if you want to look at the impact of seats on learning, you don't actually have anything. All the seats, as far as you can tell, are identical, right? You can't say anything else. But there might be some inherent difference between all of these seats that makes a difference to your learning. There might be something poking in one direction and maybe you won't be able to, you're distracted through the class. So there are various things that are possible and this typically happens with space and time. And in the example that we had, let's go back. Here, what would you have as random effects here? Yeah, position or just the identity of these. So you have the identity of these fragments. So these fragments, for all you know, all you can see cursorily is that they're all similar. You have one fragment here, you have one more there. They look pretty identical, but there are some inherent differences that might actually impact prevalence. Similarly, within each of these, you have another layer of plots, which all, as far as you can say, let's say they have the same canopy cover, they have many things that are similar. There is some inherent difference as well between them, and that is what we're trying to get at with mixed effects, right? So in addition to all of your explanatory variables, you're also adding another dimension of something you cannot explain. Yeah.
So what about things which, I mean, you're still, when you're saying you cannot explain, you're still talking about it. I mean, you're saying it could be this or it could be that. But what about something like differences in soil or some completely, like you have no clue whether it has any effect or not? So those things can also be.
It can.
Yeah.
Yeah. So as long as it's, so you have different pots, and each of the pots, you have some experiment that's going on. You have many things that you've measured, but it's possible that something inherent about each of those pots is also causing some impact, which you don't know. You have measured many things, but aside from everything that you measured, there might be something about the construction of those pots or the soil that was used that is actually causing an impact. So that is where you can bring in this random variation. But that usually this is useful when you have multiple samples within that site or within that within a particular pot. Then that will give you that's when you can actually use random effects. If you just have a single plant in every pot and you're looking at something, then you cannot do it because you actually don't have the data to look at the impact of a particular pot, right? So yeah.
Thank you. Just adding on to Uma's statement here, that I know it's like made up data, but you are dealing with fragmented and unfragmented landscapes, right? So they might have some variation among them. For example, fragmented variable would have so many additions to it, and non-fragmented would have that too.
Yeah.
So now you are adding just one variable that is different. Would you be able to add like several together?
Yes, you can. I mean, ideally, when you're designing the study, you'd want to collect a whole host of variables that you think are theoretically likely to impact your response. You might want to collect things like canopy cover, tree density, soil moisture, I don't know, whatever, anything else that you think will impact pathogens in some way, right? So all of that data will go in as fixed effects. So that can be there in a GLM without the additional M. Now, in your for the random effects, you're looking at something that beyond all this, there is something else that might also be different, but you cannot put your finger on it. So that's when you use it.
So I'm not sure how this would work, but would you be able to pair this with like distribution models? So like distribution models usually take care with like, several different variables all jammed up together, and something, would this pair? I'm just asking, I don't know.
I don't know whether distribution models have the possibility of random effects. Maybe Ashwin or somebody can tell.
Distribution models. How do distribution models compare to GLM, MMM?
How do they handle random effects, or do they have inbuilt ways to handle random effects?
Sorry, what are distribution models?
Do you want to?
Oh, species distribution models are completely different. Yeah, they're completely different. So, I mean, it depends upon the kind of model you use, but mostly you're using like Maxent or some of these kind of machine learning approaches, which actually don't use but you can actually use like logistic regression for species distributions because it's a 0-1. But generally nowadays, when you're talking about species distribution models, you're talking about these kind of machine learning approaches, which actually don't follow kind of more classical statistics. So it's a little more complex. Yeah.
It's kind of correlation.
It's completely correlation, and these are all black. Yeah, so these are all black box models, right? So you don't really know how an individual variable is affecting the outcome. They are non-linear, there are multiple, but they're very powerful for prediction. They're not very good for explanation. Yeah, so that's the difference.
So that's a good way to put it. Yeah.
Thanks.
Two mics now.
So now when we speak about random variables, before designing our experiment, we list down a set of variables which may influence it. So we test all these variables, and if they hold true, as you said, they are involved in the GLM. So when we can't explain what is causing variation, that is when we go to GLMM.
No, this is something that you need to think about right at the start. when you design the study, when you collect data, you need to have an idea of what kinds of variation there are that you actually haven't collected data to explain. And that is an important thing right at the start. So just when you design your study, you should be aware that, okay, I'm collecting all of these explanatory variables, whatever they are, but I also know that there might be some difference between these 4 different landscapes that I'm looking at, or these different things, these different sites, even within the same area, that actually you don't have an explanatory variable for.
Every study must involve GLM because anything could vary.
Exactly. That's kind of what I was getting at.
Yeah.
That there are so many things which could vary.
Yeah.
But I think the key, I don't know, maybe I'm wrong here, but the key point that you made is also that element of variation, whatever it is, should also cause intra-site variation.
Yes.
So it's not something which is differentially causing variation across sites, but that's randomness, right? Inherently, it causes variation. Is that okay to say, or Guha is frowning, so I guess not?
I think it is. So I'm not a trained statistician or anything. I'm a much more kind of practitioner, kind of statistician. So my use of random and fixed is a very pragmatic use, right? I think Ashwin is talking about a much more statistical approach to this. From a purely practical point of view, right? You can actually use, so you have to go out and collect these variables anyway. So these are variables that you have, right? You can actually use a variable as either a random effect or a fixed effect, right? So for example, you can use, for example, again, I'm very clear about this, so I use sex, not gender. So sex, you can use sex as either a fixed effect or a random effect. Now, when do I use it as a fixed versus random effect? It's a very simple criteria for me. If I want to make predictions based upon sex, so if, for example, I want to ask the question, do males have significantly higher parasite counts as compared to females? I will use it as a fixed effect. If sex is just a nuisance variable, right? I want to make predictions. I know that sex is going to be affecting, but it's a nuisance variable. I just want to control for sex. Then I will use it as a random variable. So in this sense, for example, sex is actually a bad example. So for example, if you're using sites, you have 4 sites, right? If the site itself, if you want to say, does site 1 have more parasites than site 2, then you use a fixed variable. If you are interested in asking, if I just go out and do the study all over again, and I take 4 completely new random sites, what do I really want to say? Do I really care about the identity of the sites? No, but I want to control for that inherent kind of correlation of variables within a particular site, right? So then I will use site as a random variable. So for me, at least, random versus fixed is just a question of whether it's worthwhile for me for my predictive model, or it's just a nuisance variable that I have to control. I know it's affecting, I have to control for it, but that's all I really want to. But I think that's...
I think you put it really nicely.
Yeah, so that's it's a different practical way to think about it, right? Basically it's not just some voodoo variation type of like something, right? But something which you want to make a prediction based on, and something which you have, of course, you have data for. You have to have data for it.
You have to have the data because you have to put it in the model.
Yeah.
So if you just have...
for empiricists, that's also an issue.
So, what Ashwin is saying is perfectly right. So from a broader perspective, it's perfectly right. But from a practitioner's point of view, right, you have to go out, you have to have the data to put it into a model, whether it's a random or a fixed effect. You have to have the data to put it into the model. So in that sense, as Ashwin said before, you have to know beforehand that, A, B, and C are important, and so you have to collect it whether or not you're going to use it as a random or a fixed.
So I would put it in
variable.
a slide, and I would add a little bit to that. When when you're starting off, rather than what you know is important, that's something that you can't know. I think whatever you have the theoretical understanding to say might impact this. So it's all about what you know from literature, whatever your understanding of the ecology of that system is, anything that you think can explain it. For example, so sex in this case, if there is some theory that that tells you that sex actually might drive prevalence in some way, then it makes sense to have that as an explanatory one, and then it also becomes part of the question that does actually sex impact it, or what way does it impact it. But if you suspect that there'll be some difference because of some difference in physiology, something that you don't understand about the two sexes, it makes sense to just control for that as a random effect. So
Okay, yeah, that's a
something
great
like
explanation. And I think it's really great. One of the, I think, gems that Ashwin and we've been exploring is that you have to think. Sorry. Because usually, like, I don't know, I'm an exploratory person, so I'm like, let's just see what's going on. I don't think. So I think it's really important as, people, as you're starting out projects, to actually think of all the variables. Sorry, this is my shortcoming, which I had.
Thanks, yeah. Any other questions at this stage? So this is fairly clear, this level?
What happened?
This side.
Yeah, they all look very excited. Yeah.
Yeah. So usually for there are many different packages. If you use R or if you use any other statistical software, there are many different packages that you can use to do GLMs or GLMMs. One that is used quite often is a package called LME4, and it's something that you can start exploring. We're not going to be doing anything practical today with the softwares, but you can try exploring this package called LME4, where you have a function called GLMER, which is generalized linear mixed effects, and what is it? I forgot. What is ER? Does anybody know? Okay, whatever it is, this is the function that you use to also specify random effects. And then you can have family equals binomial or Poisson, mentioning the kind of transformation that you require. Yeah. Mike.
So these are all like you should be having a basic knowledge of coding and all to do this, right? So you use like how we call command prompt, like in Windows when we type using command prompt. So that is how the interface looks like, right? When you use this. So do you have any softwares which have got graphical user interface to apply all this stuff? Because everyone does not have the background of coding.
So one thing I would say at this stage, there are some statistical softwares, but they don't have the same capability as the ones where you can actually write things down. And they don't have the flexibility to actually be able to do what you want. Now, it can seem really overwhelming, but languages like R or even Python are what are called really high-level languages. In that, all of the complexity is taken out of it. Like things like C and C++, we don't, I mean, it's impossible. I have no idea what's going on. And it's really difficult to understand. It's really difficult to find mistakes. It's really difficult to do anything there. But R and Python and MATLAB, if any of you use MATLAB, these are very high level in that a lot of the basic things are taken out of the hands of the user. And you can just with a few statements here and there, you have your data, you have different ways to store that data. And then very quickly you can reach a stage where you can actually do the statistics. And over here, you're best. Now, you're all actually very privileged to be here during the time of AI for coding. And it has simplified learning code so much, where if you have an error, you just ask your AI, like, why am I getting this error, right? Some of you have already done this. And then it will give you a reason. And then you try and fix it, it won't. Then you can say, this is not correct. Can you check again? And then the AI will do it again. And then you can use an exclamation mark saying you're upset and that it's not fixing it after a few sentences. And eventually it does. Eventually it does fix it. But that allows you also to learn. Because every time there's a nice explanation that it's going to give. It's not just going to give you code. There are wonderful explanations that are given. Do not trust what AI software say blindly. They do make up things. They tend to lie. Also, there is a yeah. So these are things we need to be really careful about. Yeah. So make sure that it's not doing something. Yeah, yeah. Very true. Yeah, yeah. Yeah, there's a really nice data set on R called Iris. And Iris is a fantastic, simple data set to just play around with and learn. It's just very simple measurements of plants in different sites. And it's something that I also typically use to teach R. So,
Yep.
right, but give it a try. It will be overwhelming at the start for anybody who hasn't used it, and I'm sure you'll pick it up soon. So yeah, so now getting deeper into fixed effects and random effects. Fixed effects are typically your treatments, different covariates for which you have some past knowledge is likely to influence the system. You have your intercept. Then you have random effects, which can be the individual host, it can be site, it can be plot, it can be sex, it can be many things where you don't actually have a reason to expect any impact, but there might be some impact. And then you have a response variable being influenced by both of these. And typically what you have, the maths would be specified in some way like this, where you have a transformation. So this is binomial data. You typically have a logit link. This is called a link. You can, you have different link functions that transform the data in particular ways. For Poisson, you have a log link. And then you have your estimates, which is whatever you can call it, A, B, C, D, or whatever, your alpha 0, alpha 1. You have that associated with covariate 1, this one with covariate 2. So this can look a little complicated, but it's actually very simple. Just like a linear model, what your GLM is going to do, or what your GLMM is going to do, is it's going to estimate these with some degree of precision. Right? It's going to give those estimates and it's going to give standard errors around those estimates. And that is actually primarily what you're looking for. Now, if these estimates are, estimated with very, very high standard errors, very large standard errors, then you cannot be confident about those estimates. Right? So you would see all of this talk about significance, right? And p-values and all of that. It's, I think you don't really need to think about that at all. You just have to think about the uncertainty around your estimate. So if your uncertainty is large, if your standard errors are large, you're going to have a higher p-value. That is where it comes from, right? The more precision you have, the lower you have that p-value. So forget about the p-value and significance, look at your estimate and the uncertainty around that estimate. What you have here, these are two, there are, you have this COV1, COV2, and you have COV1 into COV2, right? And in your model itself, you specify this slightly differently, but conceptually, you have an independent impact of your covariate 1, of let's say, you have an independent impact of something like canopy cover, you have an independent impact of soil, but you also have some kind of combined impact where for different soils, the impact of canopy cover is different, changes. So that is what is called an interaction in any regression. So that is something for you to think about. Do you expect an interaction or not? Right? Do you expect these two to completely act independently, or do you expect there to be some impact where for certain values of a particular covariate, you have the other one changing its the way it acts? Right? Does that make sense? So let's say, again, I'll use, I think sex is an easy one to use, but I don't know how appropriate it is. But you have a reason, let's say one of your questions is how amphibian sex impacts the likelihood of pathogen being present, right? And maybe there is some theoretical reason also to expect that. But what you have here, you have sex as one of the variables. Let's say you have moisture as another variable. Now you have covariate 1 is sex, covariate 2 is moisture, I mean whatever microenvironment, moisture in the immediate environment. And then you have this, would you expect an interaction here or not? The interaction here means that for one sex, the impact of soil moisture is different than for the other sex. So if you had two different lines, so let's say you have one relationship with soil moisture goes like this. Let's say this is for sex 2. For sex 1, actually the impact of soil moisture goes like this. So that estimate itself changes. The way it works is it becomes, for cov 2, it becomes cov 2 in, you can open brackets, alpha 2 plus alpha 3 into cov 1. So cov 2, that impact doesn't only depend upon, doesn't only depend upon alpha 2, it depends upon this as well. It depends upon cov 1. So when cov 1 changes, actually there is some change in the slope of this line. That is what an interaction is. So in this case, would you expect an interaction? Could the impact of soil moisture depend upon sex as well? Huh? Maybe. Okay. Possible. I mean, we're just like speculating. But yeah, there could be circumstances where this makes sense, but you'll find a really large number of real world circumstances where you expect interactions. So think carefully about interactions. This is something that when you're building your model, when you're writing it out, it's something you need to be really careful about right from the start. Do you expect one of the covariates to impact another covariate? Right? You already started out with the expectation that each of these covariates impacts the response. But do they also impact each other's impact on the response? So that is when you include interaction terms in your models, right? So, so here, now a general question. Should you use, for this particular data, we have frogs in each plot, we have multiple plots where they're sampled, we have a number of positives in each plot. If we want to use a GLM to understand the impact of certain covariates I've collected on prevalence, should you use presence absence as a response or should you use a proportion? What should you use? Sorry? And why is that? Can you talk into the mic? Yeah.
So we will be using presence absence data as our response variable because we'll be using a binomial family for this. So for that binomial function, we need a presence absence data.
So you can also use proportions in binomial models. I'll explain how.
Yeah.
So you can also do that. But is there any other conceptual reason to use presence absence over here as your response? Anyone? No, let's say that mathematically both are possible. Conceptually, in terms, ecologically, which one would you want to use over here? Your data is that for every frog, you have a 0 or a 1.
Proportion will be changed with the number of samples.
Okay.
If number is high, then proportion will be, that whatever you're telling error will be less. And
you have a denominator. Yes, that is true. But let's say that we have ways to deal with that. I'll show you how you deal with that also. But assuming you don't, like ecologically, what would you want? And under what circumstances would you want to pool everything together over here and have a fraction? Under what circumstances would you want to treat each one separately? That is the question. Presence absence means each frog or each rodent, you're treating it as a 0 or a 1. Each of them becomes a data point. If you want proportion, you're going to look at what proportion of the organisms in a particular plot actually had the pathogen, right? So under what circumstances would you use either of these? One advantage of using proportions in such a case is that it removes, it makes the model a little easier because there are going to be fewer things that impact it. But there's a very important ecological reason to use presence absence. So can anybody just say that? Yeah, anything? So, yeah.
So if you can see the tadpoles or whatever they are, it's coming exactly in the same groups of 3 in 1 out of 6 patches.
Oh, so this 3 is just representative. It's like it could be 100, it could be 50.
Across all of them? Like
No,
if it's
just
100,
it
then
could
it's...
be 100 in this, could be 25 in this, 86 in this, just
all right.
any number, how many ever you find. Let's say you put out a set of plots, or you go sampling in a particular systematic way and you find some number of frogs. The number of frogs you find in each one will be different. Now you have information per frog. You also have information aggregated across all frogs. So which of those should you use as your response? What do you lose out when you aggregate across all frogs? Frogs, yes, exactly. So the moment you have, your question involves something about the individual animal, or it involves anything to do with any data that will be lost when you aggregate across all of them, you'd rather use presence absence as your response. So if you have anything like the age of the frog, size of the frog, weight, if you expect any of these to impact prevalence, then you want to use zeros and ones as a response, and you want all the individual covariates to also figure in the model. That makes sense, right? If you don't have any of that, if you're only collecting data at the level of the plot, it doesn't really make sense to use a 0 and 1 response. If the only thing you have is at the plot level, you have tree density, canopy cover, something like that. I'm making all these up. I mean, like, when I make up covariates, I find it easier to do things related to plants for obvious reasons. But yeah, so representative. I'm sure all of you can think of much better covariates that are relevant here. So if you have things only at the plot level, if you only have things at the site level, then there is absolutely no need to use a 0 and 1 response. That's clear? Yeah? Okay. So in this case, I'm assuming that we all have information about each individual organism. We'd use presence absence with nested mixed effects. So what are nested mixed effects? The first level of mixed effects is at the site level. So these are the sites, all the big circles. The next level of random effects is at the plot level. So first, there is some inherent variation between among all the sites. Then there is some inherent variation among all of the plots within that, right? So when I showed this previous one, what we have here is actually a nested random effect. You have site, within that you have plot. So this is something that you'll again have to think about when you're writing out your models, when you're designing your study. You might want to think about the nestedness of these random effects as well. So it's clear what it means that it's nested.
What is that one, Ashwin? That one over there? What does that imply?
Thanks for asking. That means that the random effects, at least you feel that the random effects are associated with your intercept. So that's a really good question. You can also you have this alpha 0 here, right? You can also have that here. You can even have this. So you can even have one of your estimates, one of your parameters that are associated with one of your covariates. You can even expect that to be impacted by your random effects. What you have in the usual basic writing of random effects is when you expect your intercept or something that is not associated with any of your covariates to actually be randomly impacted. But there are cases when actually we'd expect these also to be impacted. Again, something to think about carefully. And I have also used that in the past, and I think many of you will find use cases where the relationship with a particular covariate, you expect that to change with your random effects. Right? So sex is a good example. Again, where if you have sex as a random effect, you might actually expect a particular relationship here to be the one where you expect that impact of the random effect. That particular one might be, I don't know, name something ecologically. moisture. So if there is a term for moisture over here, the parameter associated with that, right, and these are called parameters, there's alpha 0, alpha 1, etc. So this alpha 1 is what you might expect to see some impact of this random variation brought in by sex. You may not expect it for the intercept itself. Only the association with soil moisture you expect something to happen, but you don't know what. So you use it as a random effect. Yeah, because you have no theoretical background that gives you an expectation there to put it as a fixed effect. That's clear? Yeah.
Like fixed effect, can we put weightage also of each variable or something?
You can put weightage as well. So these models have ways to assign that also. Yeah. Usually you don't need to. Usually you need to weight your response only, not the not anything here. I'll show you how. I mean, I'll allude to weighting your response soon. So yeah. So presence absence with nested mixed effects. So this might look scary, but it is how you write out a GLMER statement. But actually, it's very straightforward. You have your presence absence, you have your this variable. So in R, data you have a kind of data type that is called a data frame. So data frame has it's like an Excel sheet. You have your columns and you have rows, and you refer to everything typically using the columns and column names. So each of these, all you need is a column that has your response. Then for each of your, every row will be every single individual data. Every individual, is it a 0 or a 1? Everything associated with that individual. Then you have all the information at the plot level also coming in that row. That individual, what information did you collect at that plot level? And that will be repeated across all the individuals of that plot. Then the site level information, which might be something like, let's say, habitat type or something, might be repeated across, will be repeated across the entire set of individuals in all of the plots within that site. Right? So that's how your data is going to be. So then you have this presence absence is a function of age, is a function of modification type, which is your blue and green, a function of tree density, and then you have your random effects, which is one with the intercept, you expect it to vary randomly with site and plot. You specify your data, you specify your binomial, I mean that it's a binomial transformation, and you specify the link function also. The link is actually the how is it being transformed mathematically. So there are different ways to deal with binomial data. So this is logit link is the usual link that is used, but you also use something called a complementary log-log link. And there are, in many of these GLMs, there are different links that you can use. Yeah. Huh.
We know which link we are supposed to use.
So that there's a lot of literature out there that specifically for your data type, there are ways to for different data types there is some theory about which links you should use. So you'll find that information online. Yeah.
There is a literature, that's fine, but how do we understand that? Like, how do we logically put it? That is most important.
Okay. No, that's a good question. A bit difficult to explain here without having a specific data type and actually looking at the way that data is distributed and so on. So for this one analysis that we've recently been doing, it's a state of India's birds. So using eBird data, and that is again we use a binomial GLMER. There actually we use a complementary log-log link for certain reasons that are inherent to that data. So if you have this question, I'll also be happy to take a look at that data and we can look at the pros and cons of both whenever that situation arises. Yeah.
Data, but I just wanted to know what is the thinking process behind applying, that's all.
It just depends upon when you do the transformation, how closely it approximates a Gaussian distribution. So eventually it all comes down to that. Yeah.
Whatever we do, the transformation, after the transformation, the data set has to look like the Gaussian.
But you won't be able to see that.
Yeah.
But there are ways to be able to do diagnostics for these GLMERs to look at both of these models and see which ones actually is working better. Yeah, which one of these transformations? There are some other things also, other choices, and I went through some of this theory to decide which of the, what would be most appropriate. I'm not sure whether I've still selected the correct one, but it felt like weighing reasons on both sides, this felt like the better choice to make. But these are not, you're not going to find big differences between these different link functions. Yeah.
Very practical. So basically, if you look at the formula, right? If you're doing like a binomial regression, your as Ashwin was mentioning, your response is kind of bounded between 0 and 1. However, your experience variables can go from -6 to plus.
Yeah.
Now the problem that you're facing now is that if you're making a prediction from this response, you want that prediction to be bounded between 0 and 1.
Yeah.
Right? So that it makes sense. Right? You don't want a proportion that goes to minus infinity or plus infinity.
Yeah.
So the logic link does that. Right? So for example, so what is the logic? Log P over 1 minus P. Right? So P is the probability of success. Right? So when P is 0, mathematicians will kill me if I say this. Right? But log of P approaches minus infinity. Right? When P is equal to 1, P over 1 minus P actually approaches infinity because the bottom becomes 0. Right? So technically what you've done is that you have taken this and converted it into a value that can go theoretically between minus infinity and plus infinity. And so that is what is linking. So that's a link.
Yeah.
So you take a response variable which is 0 and 1 and you link it to an exponent variable which can go from minus infinity to plus infinity. Right? If you're doing a Poisson regression, a Poisson goes from 0 to plus infinity. So you use a log link. Right? So basically you're again you're only interested in taking that the zeros and converting the zeros to a very like a negative number. Right? So each of these links is just basically depend upon what your response is.
Yeah. So both of these links work on binomial data. Now again, the choice between them I think is not sufficiently important. So at this stage, but yeah, you'll find reasons to choose one over the other. Yeah.
Actually, I just wanted to ask, what is quasi-binomial modeling? Sorry if I'm pronouncing it wrong, because
Yeah.
I have seen authors using it with proportion data such as prevalence data. So can you tell me, like, explain it?
Yeah, so I don't know the mathematics of quasi-binomial, but I think it is typically used when you have lots of zeros or something, right? When you have zero inflated data, like more zeros than ones. So similarly, I think there's also quasi-Poisson, no? Or negative binomial. There are many of these that I think practically, I don't know the maths behind it, but practically they're used when your data tends to be zero inflated. So that's when some of the more conventional techniques fail. So then you move to some of these other models. But these are actually very robust modeling techniques that are not impacted so much even by zero inflation. So,
Thank you.
yeah. With zero inflation means you have a lot of zeros and very few ones. Or you have a lot of zeros and very few ones and twos and threes in a Poisson in some count data. Yeah. So these, so there are certain optimizers you can use. These are all things that make your model a little faster. You can play around with these. But the really important part is this. This is where you actually ecologically write out your GLMER.
So all those things are just to improve your application?
Yeah. Just to make things faster. Yeah. Because otherwise they tend to do a number of diagnostic steps. They go through, they keep, it simulates multiple times to see if the same thing is coming out. And I think there are various diagnostics that are sometimes not required. So it takes a lot of time. Then, yeah. So after that, after you have the model, after you get the results, after you get the summary of the results, you're going to be viewing the results on the transformed scale. right? You're going to be viewing it not in the with the estimates actually being immediately translatable to the 0 and 1, right? It's not going to be that you put your covariate values back in whatever results you get. You're not going to get a value between 0 and 1. You're actually going to get something from -infinity to +infinity, because that is the scale at which now your results are going to be given. So you need to be careful about interpretation at that stage. You cannot say that with one change in the, let's say we have here modification type, and what is associated with modification type is minus, let's say some number minus 0.4, right? Now you cannot say that with if modification goes from, if the type goes from fragmented to unfragmented, the actual change is 0.4. That is on the transform scale. You need to be able to predict onto the onto your actual real scale. And JLMER and all of these this code makes it quite easy. There are ways to, there's a predict function. Using the predict function, you can, for certain covariate values, you can get a predicted response value, right? So you put in the data for, like, let's say for fragmented, unfragmented, tree density going from 1 to 10, from 1 to 100, all of these numbers you put in a dummy data frame, and then you can use that predict function to get a response on the actual real scale, untransformed, back transformed scale. So also check the mean of each estimate in the model results. You do summary of it and you'll get the model results. Check the estimates, check how much precision there is. And I didn't actually have a data set to do this with, so I haven't shown this, the model results itself here. But this is what you want to do. And the moment you have any data, immediately try and put it in this format and start looking at the model results. You have your estimates, you have your standard error, see what you can interpret from that, right? Smaller the standard error, the more confident you are in those estimates. Then predict means and standard errors using the predict function to be able to actually get values that you can plot on a graph. For different circumstances, you have a mean, you have uncertainty for and values between 0 and 1. That's what you're going to be predicting there. With the GLMER, with the GLM, it's very easy to use predict and get confidence intervals or standard errors. With the GLMER, you might have to use a function called bootmer. That it would be, you can't immediately calculate it, you actually have to bootstrap. We spoke about bootstrapping earlier, no? That you're resampling and you're constantly getting a mean value. Similarly, we'll have to do this over here, and the model therefore will repeat itself 1,000 times or 10,000 times, and each time you're going to get a mean value, mean predicted value, and with that you're going to get your mean and uncertainty. And this is something that I would say that you need to practically try. And once you start exploring the code and make some mistakes, you'll figure out how to use all of these and even understand them conceptually better. And finally, you might want to, I couldn't think of anything else, but but you want to visualize everything. Finally, for your audience, right, to be able to communicate anything with any reader, you cannot just put these model results out there. Nobody is going to be able to interpret it, right? It's just a bunch of numbers that nobody can really understand. You can, maybe it'll tell you something about what is estimated with precision, what is not, what you can be confident about. Like you can say maybe the estimate associated with tree density is something that is quite precise. So you can say that this relationship with tree density we can be fairly confident about. But some of the others might have larger standard errors, so you're not confident about that. But to actually see how this is reflecting in your fit itself, you have your data points, you have this model, and actually in many cases these models can even make mistakes, and you might actually have a prediction that is way off what your data actually shows, right? You want something to be able to actually explain your data. So it's useful to predict and back transform and actually plot your model results alongside the data, and that is quite important. This is important in any, whenever you do any kind of modeling, wherever there is like an opaque box where you don't know what is going on inside, it's useful to bring this out, see how it's fitting with your data points themselves, and let that be the visualization that goes back to the reader, so that they can look at this. Now there's a relationship between predicted prevalence as a function of canopy cover, and I think Uma will be happy because I've completely changed everything here. It's just the same graph. So, so you have inverse of canopy cover going from 0 to 1. As canopy cover increases, the model says that there is decreases, there is increased prevalence. So in more open areas and slightly more disturbed areas, maybe there is increased prevalence, and it looks like this particular model fit. What you have in the middle is the mean line. For different values of canopy cover, values of prevalence have been predicted. Similarly, for each of these values, you also have an uncertainty, standard error, or a confidence interval that is predicted. That is the what's it called? The translucent ribbon. And the middle line is the mean line. So then the reader can look at this and see whether they think this makes sense or doesn't make sense. Because this is what your model has told you. So this is what you want to end with at all times. Now, some of the later parts involve coding, and it's useful to start getting used to it because it's actually easier than everybody thinks it is. So that's all I have. I feel I must have taken 1.5 hours. Do I have time? I'm on time? So we'll have some questions now and have 5 minutes. Yeah. Any questions? Was this, you got a broad idea of regressions? What is the linear regression? What is the generalized linear model?
Data is going with linear regression also.
If
data is following the linear regression also, then also we need to do GLMM, or we don't require, na? If it's perfectly fitting with that, then we don't require, I think.
So if your data actually, it's useful. So this is actually a good point. Right in the beginning, it's useful sometimes to explore your data, to plot some scatter plots and see what it's looking like. For binomial, for 0 and 1 data, you cannot directly use a linear regression anyway. But for other kinds of data, like the length of a leaf or the size of a weight of a table or anything like that, if you actually find that your scatter plot is non-linear, or it looks like it is showing some kind of relationship that is not linear, you might want to consider not using a linear regression because it doesn't make sense. You're actually trying to fit a straight line into something that actually is not a linear relationship. And this happens a lot in ecology. We try and put a straight line through everything, but very often don't have a straight line.
This data also, if I made the straight line, it will look like it's fit.
Yeah, but
how I'll know that I need to do GLMM and all?
Yeah, so in this case, there are 2 reasons why you shouldn't do it. One is because it's 0 and 1 data, which anyway, the moment you fit a straight line, you're assuming that it can take any value. It can take a value. So you see what happens with this is that this value can never go below 0 and it can never go above 1. It's bounded at 0 and 1. So the moment you have a straight line, what happens? You're cutting through the entire thing and then it goes below 0 and it goes above 1. As your values become larger, it just keeps going up. Right? So that is not appropriate conceptually also. So these are some of the things you need to think about before deciding whether it's linear or non-linear. Yeah.
If the data was about a number of viruses within a rodent or the viral load of a fungus on a frog or something like that, we can use linear regression directly. Can we?
Assuming, 2 things. Assuming that theoretically you expect it to scale linearly with whatever response you have. That is one thing. The second thing is, do you, what are the bounds of this data? So if you again have, if it's count data, so if it's again count data, you might want to put it on a scatter plot, see whether sometimes you might actually have a relationship. All the scatter might be somewhat linear, in which case you can at times also use a linear regression for count data, but typically because it is bound, right, in the beginning, it cannot go below 0. You're going to have many of them near 0. So usually it's going to, there's going to be a need to transform it. So even for this kind of count data, I would say mostly you'll use a Poisson regression because of the bounding, largely.
Sir, actually, we'll be using variance inflation factor for to see a multicollinearity of different variables. So that WIF can be used for random effects. Is that correct?
I didn't.
Yeah.
Yeah, I don't know how that where random effects come in here, but with fixed effects, it's useful to look at whether any of your variables are correlated. If they're correlated and if they're like, let's say, if one of them explains the other, it doesn't make sense to have both. You might want to choose the more important factor to keep in your model. You don't want everything. Like for example, for humans, if you have measurements of even rodents, you might have a measurement that is the weight of a rodent. You might also have a measurement that is the height of a rodent or something, right? But you might have weight and height correlated quite strongly. So you just need any one of them. The one that actually you expect will make a difference, which might be the weight. The weight might have a bigger bearing than the height. So that will be what you want to use. But yes, looking at how all your variables are correlated with each other is an important step before deciding what you want to keep in your models. Yeah.
Yeah, you spoke about GLMs and GLMMs. One other thing that came across in Ansil's slide in the morning was GAMs. So can you briefly touch upon what that is and how different it is from these two?
Right. So I don't know the maths behind a GAM. Does anybody know the maths behind? It's a generalized additive model, and yeah. But I can tell you practically when you think of using a GAM, it's when you actually don't have you have a relationship that seems to follow some kind of funny. There's some kind of funny relationship out there, and you cannot think of it fitting into any of your conventional GLM type relationships. For example, when here you have a relationship that goes like this and then plateaus again near one. So you have it going like that. You have certain, typically all of these relationships are, what are they called? When you only have a single peak. They're either monotonically increasing or decreasing. You don't have it coming down and going up. Whenever you have data that is following a curve like this, it's going up, coming down, it has multiple peaks and troughs.
Is it called unimodal or bimodal or something like
probably, yeah. So if you have in something that is, even unimodal can go up and down. Even in that case, none of these, this framework will not work. That's when you want a GAM where you can make it fit all sorts of funny relationships. I don't know the maths behind it though, but that's when you use a GAM. Yeah, you have your data just going like this. Seasonal data.
Pathogen prevalence. Yeah, again, I don't know how the model works, but probably like you remember the coronavirus prevalence changes over time, the plot we saw. Such data, maybe we can use these additive models, something.
Yeah.
But probably in the coming days, some of the people who uses the additive models are going to be here. Maybe we can ask them how the models work and how do you do such things, at what context you use that things like that. Ma? Cliff probably be using.
Oh, that's perfect then. Yeah.
It's just with all the random effects and everything. I have done lots of GLMs, GLMMs and everything. But most of the time, actually, it's the simplest model, which works really well. Because disease data, most of the time, is very skewed. I mean, you may be recording lots of things in the field, hundreds of rodents and everything, but your parasite prevalence might be very, very low. So those random effects may not work or model may not converge or anything. So, I mean, I think what Ashwin has showed you is a very fantastic and very simple model and good. Assuming there's a good prevalence, maybe 50% or something he was showing, or more than that. But I just want to say that it's sometimes random effect doesn't think. If you think, you may think would be nice to pull it off, but it may not work. So
yeah. There are many practicality issues that you'll bump into as you start dealing with the data. And just actually before I close, I mentioned these weights when you're looking at proportion data. When do you use proportion data? Typically when you don't have either individual information or you have things like experiments. You have a bunch of them, you put a bunch of rats together and you want to see whether how many die from the pathogen or something. And so you have a proportion, proportion that has died. You don't know which individual, what has happened to each individual, you just know that 7 out of 10 have died. In that case, you would want that to be a response. You have a number of explanatory variables associated with that, but here you want your response to be the proportion. When you have your response being the proportion, it's really important that you have weights. So this is an easy thing to bring into any GLM or GLMER, where you can specify the weight, which is the number of trials that went into that proportion. So for example, a proportion of 0.7 out of 10 trials is very different from 0.7 with 100 different trials. So 10 rodents and 7 dead is very different from 70 out of 100. So the way, so the model also incorporates that while looking at how confident it can be with the estimates. So it's very important to add weights when you're dealing with proportions. When you're looking at individuals, you have zeros and ones, you've removed that complication out of the picture. I would also say, as a general piece of advice, don't throw in 100 variables into every model and then compare using AICs and keep moving down. It's much better to start with the ecologically most meaningful model after deciding which few variables are actually make sense theoretically, and to stick to those. You don't drop anything, you don't add anything. That is what you have as your starting model, and that will also give you that will lead you through I think an important process of thinking in the beginning, where you actually think very carefully about what you want to collect, what your different covariates are, and then you're going to stick to actually the most ecologically meaningful model right from the beginning to the end. Yeah, and then I had this here, I forgot about this slide, but yeah, check whether your variables are correlated. But yeah, I think this is it, yes. So, thanks. Yeah.
Thank you so much, Ashwin.
Yeah.